Hello Warriors, this is Miss Lemons, and today I'm going to be walking you through your base setup for the seed germination lab that we're about to do. So keep in mind that this setup is for your control group and is going to be what you alter one variable for each of your treatment groups. So again, this is our control group setup. So I've gone ahead and I have listed all of the materials that you're going to be given at the beginning of your lab. You get a paper towel from the dispensers, a plastic bag, just a sandwich size. You'll get several plastic bags, but this is just for our control group. You'll have a 10 milliliter graduated cylinder, so the teeny tiny one, a bottle of hydrogen peroxide solution that we've already made for you. So if you guys are familiar, like this is what hydrogen peroxide usually comes in, the big brown bottles at the grocery store. You typically use it for like wound care. This is actually pretty great for germination of seeds, so we are going to have a pre-made solution for you. And then we'll have, of course, the radish seeds, about 40, give or take. Permanent marker so that you can actually write down your group names, and then of course tape for labeling. So the first thing that we're going to do is take a look at our radish seeds. So we're going to be placing 10 seeds in each of our treatment groups. And normally you could just like dump these in, but some of these seeds are better than others. So if you look at some of the ones that I'm picking out here, some of them are broken or they have this like light skin on them that's showing that the seed coat has actually been burst or broken. So we really want to try and pick seeds that are nice. So you can see here, this is kind of like broken open on one side. Let's try this here. So you can kind of see this one's kind of broken. This one also is kind of split, right? We don't want that. This one's not even a full seed. This is just like the inside of the seed. And then again, this is a portion, and then I have no idea what happened to this one. Okay, so we really want to make sure that we're picking out nice looking seeds. Okay, so the ones that I'm thinking of here are like the ones that are nice and brown all the way around. They don't look like deformed at all. So we're going to pick 10 really nice seeds. Okay, so I picked out my 10 really lovely seeds. They look nice. They are not damaged. So these are my 10 seeds. Every single one of your treatment groups is also going to get 10 seeds. So we have our paper towel. And what we're going to do next is add our hydrogen peroxide solution. Before we do that, though, we're going to fold our paper towel so that we kind of wrap the seeds in the paper towel like a little envelope. So I'm just going to fold it over a few times here like this. And then, of course, we want to make sure we fold it this way, too. Make sure there's no seeds getting out. So we're going to fold it this way. And then I'm going to fold it also under like this. How you fold it doesn't really matter. You just don't want the seeds to fall out of the little envelope that you're creating. Okay, so next we're going to be adding our solution. So to add our solution, we are going to be using the 10 milliliter graduated cylinder, and we are going to measure out 6 milliliters of the solution into our grad cylinders. So you want to make sure that you're careful when you're pouring in your hydrogen peroxide solution. And if you spill, just make sure you wipe it up. All right. Just a quick reminder, too, is that when we measure this, you need to make sure you're reading under the meniscus. So I'm going to go ahead and fill this up and show you exactly what I mean. Okay, so I've gone ahead and added about 6 milliliters of the solution into my grad cylinder. And keep in mind that you want to make sure that we're measuring on a level surface. I'm not doing that right now because I'm showing this to you. But you'll also notice that we have that bubble. That's called the meniscus. So we want to make sure that we are taking our measurement from under the bubble. So you can see that I'm actually slightly under 6 milliliters. That's okay. If you're like a little bit above or a little bit below, that's totally fine. But we definitely want to keep it right around 6 milliliters. Okay, so now we can go ahead and add this to our seeds. Okay, so I'm just going to carefully pour this in onto my little envelope that I've created. Just make sure all of the paper towel is nice and damp. You see that's kind of soaked through. Okay, and now we're going to add this into, or put this into our plastic bag so that it doesn't dry out. And then we'll also label it. All right, so I went ahead and I've added my seeds to my bag. I'm going to go ahead and add a label to it. And on this label, you are going to put your group initials, your class hour, and then you're also going to put the treatment group, so which test group you're in. So I'm going to write test group because I have enough room. Test group. So for this bag, I would probably write that this is my control group. Okay, so let me show you what an example label would look like. All right, so now we have our label, me, Mr. Brandt, 4th hour, control group. Okay, so I also created another one for a test group. So for instance, this is my test group that's only getting 2 hours of sunlight, and my control group is going to get the 8 hours of sunlight that they normally get. So notice that these look pretty much the same, and the only thing that I've changed between the two of them is this has 2 hours of light, whereas this is going to get the normal 8 hours of light. So you have to think about the way that you are going to be manipulating one variable when you set up each of your baggies. All right, go ahead and get started.